Welcome, and thank you so much for joining us on this custom-tailored deep dive. We're really thrilled to have you with us today.
Yeah, absolutely. Thanks for being here.
The goal for this session is, well, we want to take a pretty formidable stack of incredibly specific, highly scientific sources and basically distill them down into those brilliant, mind-expanding moments of discovery for you.
Exactly. The intention is to do all the heavy lifting, synthesizing years of field research and lab analysis into a cohesive narrative so you don't
Yeah.
have to wade through the dense academic formatting.
Right, we're getting straight to the good stuff. And the stack of literature we have for you today is actually
It
remarkable.
really is.
We're pulling from a wide array of materials. We've got comprehensive foraging guides, foundational encyclopedia entries.
Yep, and an incredibly detailed 2025 taxonomy update by mycologist Wolfgang Klofak and Irmgard Krasai-Grailhuber.
Which is a massive paper. And we're pairing that with a highly technical 2021 study published in the journal Microorganisms.
That last paper is incredible. It's going to take us deep into the temperate forests of central Chile to look at some biological interactions that are, almost alien in their complexity.
Yeah, it's a fascinating cross-section of science. We're starting with historical botanical illustrations from the late 1700s and pushing all the way through to modern molecular phylogeny.
Basically using DNA barcoding to redraw the family tree of the fungal kingdom.
So just to give you a roadmap for this deep dive, our primary subject is the wild, entirely hidden double life of a fungus known commonly as the Peppery Bolete, or, if we're being biological about it, Chalciporus piperatus.
Now, I realize that probably sounds like we are dedicating our time to a simple, unassuming little mushroom you might just casually step over during a hike.
Right. But let me set the stage for you here. What appears to be a very basic forest fungus is actually the epicenter of a massive global saga.
It really is.
We're going to be exploring the absorption of toxic heavy metals, global migrations that cross oceans alongside the timber trade,
Literal chemical warfare.
literal chemical warfare on a microscopic level, and a network of parasitic espionage that operates right beneath the soil.
The deeper you look into this single organism, the more the traditional boundaries of biology they begin to break down.
Yeah.
You start to see how interconnected, highly specialized, and ruthlessly competitive these ecosystems are. It completely forces a reevaluation of what we assume is happening in the dirt under our feet.
So let's start by unmasking this forest enigma. The Peppery Bolete has actually been on the scientific radar for a surprisingly long time, hasn't it?
It has. It was first officially described way back in 1790 by a French mycologist named Pierre Bouillard.
1790?
Yeah, he originally named it Boletus piperatus.
Yeah.
And just a few years later, in 1797, it was immortalized in a botanical illustration by James Sowerby.
Which, if you think about what science was in the 1790s, before photography, before genetic sequencing,
Oh, entirely.
those hand-drawn illustrations and physical descriptions, that was the absolute bleeding edge of biological cataloging.
And those early illustrations were crucial because they established the morphological baseline for the species. For over a century, this mushroom just sat in the massive catch-all genus of Boletus.
Right.
But as mycologists developed better tools, including early microscopy, they started noticing physiological differences that they just couldn't ignore anymore.
So it got moved.
Exactly. By 1908, another French mycologist, Frédéric Bataille, recognized that this specific mushroom, along with a few close relatives, possessed traits that were distinct enough to warrant pulling it out of the Boletus genus entirely.
And he made it the type species of a newly circumscribed genus.
Chalciporus, yes.
And the etymology of that new name is basically a cheat code for identifying it in the wild, right? Like if you break down the Latin and Greek, it tells you exactly what to look for.
The nomenclature is highly descriptive, yeah. The genus name Chalciporus is a hybrid of two roots. You have the Greek word chalkos, which translates to copper,
Copper, okay.
and porus, referring to the pores on the underside of the mushroom. And then the species name, piperatus, is just straightforward Latin for peppery.
So you have a copper-pored peppery mushroom.
Exactly.
So if you're walking through the woods and you want to actually spot this thing, what are you looking for? Let's translate those field guide dimensions into something visual for you listening.
Sure.
This isn't a massive plate-sized mushroom, right?
No, not at all. It's considered a smaller bolete. The cap is generally going to be the size of a small coin, up to maybe the size of a drink coaster at its absolute largest.
Okay, so pretty small.
Very small. When it first pushes out of the soil, the cap is convex, like a little dome. But as it matures, it flattens out, and the edges often warp into this really distinctive wavy margin.
And the color is very earthy.
Very much so. It leans heavily into earth tones. You'll see a range from a pale sandy brown all the way to a deep, rich cinnamon brown, and it frequently carries a reddish undertone.
But the texture changes too, depending on the weather.
Right. The tactile experience of the cap changes dynamically with the ambient humidity. So during a dry spell, the cap appears shiny, almost as if it's been polished.
Like it has a little wax coating on it.
Yeah. But introduce rain or high humidity, and the surface becomes distinctly greasy or slightly sticky to the touch.
Okay, but the real giveaway, the feature that earns it that Chalciporus name, is hiding underneath the cap.
The pores.
Right, because most people are used to standard button mushrooms from the grocery store, and those have those thin, paper-like gills underneath. But boletes operate on a totally different architecture.
They do. They have a spongy layer of vertical tubes that end in tiny holes or pores. And on the Peppery Bolete, these pores are relatively large, they're angular, and they boast this striking rusty orange to dark reddish-brown color.
Those are the characteristic copper pores.
Yes, and they hold a really crucial diagnostic secret.
This is the bruising thing, right?
Exactly. Many boletes are famous for their bruising reaction. If you press your thumb into the pores of certain bolete species, the flesh will rapidly oxidize. It turns this vivid, almost alien blue within seconds.
It looks like magic when you do it in the woods.
It really does. But the Peppery Bolete lacks this dramatic chemical reaction. If you bruise or slice the copper pores of Chalciporus piperatus, they do not change color significantly.
So no blue.
No blue. They might stain a slightly darker brown, but the complete absence of that rapid blue shift is a major identifier for foragers.
Okay, so moving down the mushroom, the stem is fairly slender, usually about the thickness of a pencil or a marker, and it carries that same cinnamon brown color as the cap. But the coolest visual detail to me is right at the soil line.
The base.
Yes. As the stem meets the dirt, it aggressively tapers into this incredibly vibrant chrome yellow base. It literally looks like it was dipped in neon paint.
That chrome yellow coloration is the basal mycelium. That's the vegetative part of the fungus extending down into the substrate.
And if we move from the macro view to the microscopic view, the diagnostic traits become even more refined, don't they?
They do. The spores of this fungus, which will actually leave a cinnamon brown print if you drop them onto a piece of paper, are described morphologically as subfusiform.
Subfusiform. Okay, let's paint a picture of that for everyone instead of just reading the micrometer measurements. What does subfusiform look like?
Essentially, they're spindle-shaped. They taper smoothly at both ends. In the mycological literature, they are frequently compared to the shape of microscopic cigars.
Microscopic cigars, got it.
And crucially, they are entirely smooth. They lack any ornamentation or warts.
Which becomes a very important distinction later on in our discussion.
Very important, yes, when we look at the molds that attack them later.
Right. So nature rarely produces exact carbon copies, so it's not surprising that there are a few documented variations of the Peppery Bolete out there.
Yes.
There's a rare European variety known as Hypocriscius, and instead of the classic rusty orange pores, this variety features bright yellow tubes and pores.
Which caused quite a bit of confusion. For a long time, there was this quiet debate in the mycological community about whether this was just a weird color mutation or an entirely different species altogether.
Because resolving those debates used to rely entirely on just looking at them, which is inherently subjective.
Exactly. Visual observation only gets you so far.
Right.
But the 2025 taxonomy update by Klofak and Kreisai Grailhuber utilized a much more definitive tool. They used molecular genetics.
Right.
They successfully sequenced the ITS region from the type specimen of Hypocriscius.
Let's briefly contextualize the ITS region for you listening because it pops up a lot in modern biology. It stands for the internal transcribed spacer.
You can think of it as a highly conserved genetic barcode.
Right, because this specific sequence of DNA mutates at a very predictable rate. Scientists can read it to see exactly how closely related two organisms are. It completely cuts through all the confusion of superficial physical appearances.
And by analyzing that genetic barcode, the 2025 paper definitively proved that Hypocriscius is not a standalone species.
It's just a color swap.
Basically, yes, it is firmly nested within the genetic profile of Chalciporus piperatus. So the molecular data put a century-old taxonomic debate to rest. It confirmed it as a true variety.
They also note another variety, Amaryllis, which has pinker pores and a distinctly bitter taste rather than peppery. But the discovery that absolutely blew my mind from that 2025 paper was actually a finding from 2020.
The new species.
Yes, a completely new species named Chalciporus pseudopeperatus.
This discovery serves as a really vital reminder of the limitations of historical taxonomy. You see, based on broad morphological similarities, basically, mushrooms that generally look like a peppery bolete, we just assumed Chalciporus peperatus was a single, globally distributed species.
Right, we thought it was everywhere.
But the Klofack and Creasy-Grailhuber update stresses that this is likely an illusion.
It's a case of mistaken identity on a massive global scale. We have these mushrooms masquerading under the piperatus name in places like Australia, North America, and across Europe.
And what we thought was one cosmopolitan mushroom is almost certainly a complex of several undescribed look-alike species that we just haven't officially genetically cataloged yet.
That's wild. Until we run the DNA barcoding on specimens from all these disparate geographic regions, we can't be entirely sure of the true boundaries of the species.
It really highlights how much hidden diversity still exists in heavily studied environments.
Which means if you are out foraging, that visual similarity is something you have to be very, very careful with. You do not want to mix the peppery bolete up with some of its less friendly neighbors.
Absolutely not. For instance, a novice might confuse it with Soilellus luridus, but that mushroom is significantly larger. It features a distinct net-like pattern on its stem, and critically, its pores rapidly turn blue when bruised.
Which the peppery bolete does not do.
Exactly. There is also potential danger of confusing it with Rubroboleta satanas, ominously known as Satan's bolete.
That name alone should be a warning.
It is highly toxic. However, Satan's bolete has a pale, almost whitish cap, vibrant red pores when it's mature, and its inner flesh turns pale blue when you cut it open.
Okay, good to know.
You might also encounter the suede bolete, Xerocomus subtomentosus. This one is actually edible and looks quite similar when it's aged or dried, but it completely lacks the defining trait of the peppery bolete.
The taste.
Right. It completely lacks that intense peppery taste.
Which brings us nicely to the culinary oddities of this mushroom. If you slice into a fresh peppery bolete, you find this firm yellow flesh. And just under the cap surface skin, the cuticle, there is a striking, thin red band.
And the flavor profile is aggressive.
Aggressive is a good word for it.
The common name is incredibly accurate. If you taste a tiny fragment of the raw flesh in the field, which is a common practice for identification purposes, the flavor is remarkably hot.
Like how hot?
It mimics the acrid burning sensation of heavily cracked black pepper. It is an intense chemical reaction on your palate.
It's such a potent flavor that the legendary Italian chef Antonio Carluccio specifically included it in his writings on mushrooms.
He did, but with a warning.
Right. He strongly advised against ever eating it as a standalone dish. Instead, he proposed using it strictly as a condiment, like adding a few slices to a pan of blander mushrooms just to impart that peppery heat.
Which is fascinating, because utilizing a wild mushroom primarily as a spice is a very rare culinary application. However, any discussion of consuming this fungus requires a serious caveat.
Yes, disclaimer time.
The foraging guides and scientific literature explicitly note that the raw mushroom contains gastric toxins. Many regional guides actually classify it outright as inedible. If it's going to be consumed, it must be thoroughly cooked to neutralize those toxins and prevent severe gastrointestinal distress.
But the wild culinary paradox of the peppery bolete is that the heat is incredibly fragile.
It's a catch-22.
Exactly. If you cook the mushroom thoroughly to ensure it is safe to eat, the peppery flavor completely evaporates. The heat is destroyed by the cooking process.
It vanishes.
So you might think, okay, I'll just dry it out and grind it up into a powder and use it as a literal pepper substitute. But the drying and powdering process destroys the flavor profile even faster.
It's as if the mushroom evolved this brilliant culinary defense mechanism that completely frustrates any attempt for us to utilize it.
The biochemistry responsible for that fragile heat is fascinating, though. The specific compound generating the peppery bitter taste is known as chalciporone.
Right. And chalciporone isn't the only unique chemical this fungus is manufacturing. We mentioned earlier that incredibly vibrant chrome yellow mycelium at the base of the stem.
The neon paint.
Right. The pigment creating that neon yellow is called sclerositrin.
And sclerositrin is an intriguing molecule because of where else it appears in nature. It is the exact same pigment found in a completely unrelated fungus, the common earthball, Scleroderma citrinum.
It's a phenomenal example of convergent evolution at the chemical level. You have
Yeah.
two entirely different fungi arriving at the exact same complex pigment independently.
Nature is amazing.
From a biosynthetic standpoint, the chemical factory inside the peppery bolete is highly active. Compounds like calciporone, sclerositrin, and other pigments found within its tissues, like calcitrin and norbathione A, they're all derived from a foundational base compound called xerochromic acid.
So the organism is basically utilizing this single acidic base to synthesize a whole suite of specialized chemical tools.
Exactly, it's highly efficient.
And those chemical tools actually have historical applications outside the kitchen. Because of those potent pigments, the peppery bolete can be used to dye fabrics.
That's right.
Depending on the mordant you use, the chemical binding agent that sets the dye into the fibers of the cloth, you can extract a surprising range of colors, from vivid yellows and oranges to deep greenish browns.
It's remarkably versatile. But if we extrapolate this intense chemical capability to its environmental interactions, we uncover a far more profound and frankly alarming biological trait.
This is where things get really weird.
The literature details a field study conducted in heavily industrialized regions of Czechia and Slovakia. They were specifically targeting areas contaminated by historical lead smelters, old mine dumps, and slag heaps.
So the soil in these areas is catastrophically polluted. We are talking about landscapes utterly saturated with heavy metals.
Very toxic environments.
And thriving right in the middle of this toxic burt is our tiny peppery bolete, acting like a biological sponge.
The data regarding its heavy metal accumulation is staggering. The researchers discovered that Chalciporus piperatus actively bioaccumulates the element antimony at unprecedented levels.
Antimony.
Yes. In one sample collected near these industrial sites, the fungal tissue contained 1,423 milligrams of antimony per kilogram of dried mushroom.
Okay, to help you visualize that, think about the other fungi trying to survive in that exact same toxic environment. How much antimony were they absorbing?
A negligible fraction by comparison. Whether the researchers looked at saprobic fungi, which break down dead organic matter, or mycorrhizal fungi attached to tree roots, the levels were an order of magnitude lower.
So the peppery bolete isn't just passively existing in polluted soil. It is aggressively pulling this specific, highly toxic heavy metal out of the environment and packing it into its own cellular structure.
He is pulling it straight out of the ground.
And antimony is no joke. It's highly toxic to biological life. So the obvious question here is, how is this tiny fungus not just melting itself from the inside out? It's pulling up 1,000 milligrams of poison. What is the survival mechanism?
Well, the exact biological mechanism remains a subject of intense research, but the leading hypothesis brings us back to those complex organic acids we discussed earlier.
The xerochromic acid.
Exactly. It is highly probable that compounds like xerochromic acid or its biosynthetic derivatives are acting as chelating agents.
Let's define chelation for a second.
Chelation is a process where an organic molecule essentially wraps around a heavy metal ion. It binds to it and neutralizes its immediate toxicity, preventing it from wreaking havoc on the organism's cellular machinery.
So the mushroom is isolating the toxic antimony in these little chemical straitjackets.
That's a great way to put it.
And that allows it to survive in soil that would kill almost anything else. If scientists can fully reverse engineer how the peppery bolete does this, the applications for bioremediation, using living organisms to clean up human-made industrial disasters, could be revolutionary.
It absolutely could be. It highlights a crucial point in modern biology. The solutions to some of our most complex environmental challenges might already exist right now in the biochemistry of seemingly unremarkable woodland fungi.
But if you think its ability to eat toxic waste is strange, we need to look at how this mushroom survives on a day-to-day basis in a normal forest. Because for a very long time, the entire scientific community had its ecological role completely backwards.
They really did.
Yeah.
For decades, the prevailing assumption in mycological textbooks was that Chalciporus piperatus was an ectomycorrhizal fungus.
Ectomycorrhizal. Let's break that down, because it's a foundational concept in forest ecology.
Right.
It refers to a symbiotic, mutually beneficial relationship between a fungus and the root system of a plant, typically a tree. The fungus weaves its mycelium around the tree's roots, massively expanding the root network's surface area to absorb water and essential minerals from the soil.
And in return, the tree, utilizing photosynthesis, creates sugars and carbohydrates, which it actively feeds down into the root system to sustain the fungus.
It's a massive cooperative underground economy, and almost all boletes operate this way.
Because the peppery bolete possesses the classic morphology of a bolete, And because foragers consistently found it growing in mixed woodlands, often associating with pines, birches, spruces, and beech trees, mycologists quite logically assumed it was plugging into that exact same symbiotic tree network.
It looked like a team player, so they assumed it was one.
Exactly. But the data refused to cooperate with the assumption.
Yeah, researchers kept trying to prove the symbiotic link, and they kept failing. They conducted numerous in vitro synthesis studies.
Basically, they took tree saplings and the peppery bolete, put them together in highly controlled, sterile laboratory environments, and just waited for them to form that root connection.
And it never happened. The fungus showed absolutely zero interest in bonding with the trees.
None whatsoever. So when visual observation failed, the scientific method shifted to chemical tracing. Specifically, they utilized carbon-13 and nitrogen-15 isotopic fractionation tests.
Okay, let's break down isotopic testing because this is fascinating. This is essentially forensic accounting for biology, right?
That is an excellent analogy. Isotopes are variations of chemical elements that have different atomic weights. In any ecosystem, different organisms will process heavier isotopes like carbon-13 or nitrogen-15 slightly differently based on their specific metabolic pathways.
So by measuring the ratio of these heavy and light isotopes in a tissue sample, scientists can track exactly where an organism is getting its carbon.
They can track its exact food source.
So if the peppery bolete was a true ectomycorrhizal partner, its isotopic fingerprint should closely match the isotopic fingerprint of the host tree it's supposedly receiving all its sugars from.
Precisely. The carbon signature should map directly back to the photosynthetic canopy of the surrounding trees. But when they ran the mass spectrometry on the peppery bolete, the isotopic signatures completely diverged from the local flora.
Completely different.
The data provided irrefutable proof. This fungus was not engaging in a symbiotic relationship with any plant life.
Which presents a massive biological mystery. If it's not trading minerals for tree sugars, and it's not a simple saprobe just passively eating dead leaves on the forest floor, how is it getting the massive amount of energy required to build its fruiting bodies?
The plot twist is brilliant. Chalciporus piperatus is now strongly suspected by the mycological community to be a mycoparasite.
A mycoparasite. Instead of a cooperative partner, it is an underground thief.
That's exactly what it is. A mycoparasite is a fungus that derives its nutrients by parasitizing the mycelial networks of other fungi. It bypasses the trees entirely and taps directly into the nutrient streams established by other mushrooms.
And the target it chose to parasitize is arguably the most famous mushroom on the planet. The Fly Agaric, Amanita muscaria.
The iconic red mushroom with the white spots.
The one from every fairy tale, every video game. The peppery bolete is secretly weaving its way into the Fly Agaric's root system and siphoning off its life force.
Recognizing this parasitic dynamic completely reconstructs our understanding of the peppery bolete's ecology.
Ah.
And more importantly, it unlocks the mystery of its incredible global distribution.
Right.
Because this is a fungus native to the mixed woodlands of the Northern Hemisphere, Europe and North America. Yet today, it is found thriving across the Southern Hemisphere.
It's been documented in Brazil, in New Zealand, down in Chile. How does a tiny forest mushroom cross the Atlantic and Pacific Oceans? It didn't blow on the wind.
No, it hitched a ride on human industry.
The global timber trade.
Yes. As nations in the Southern Hemisphere sought to establish fast-growing, highly profitable timber industries, they imported millions of exotic pine saplings from the north. They introduced species like the loblolly pine, Pinus taeda, to the foils of Brazil.
And they brought the Monterey pine, Pinus radiata, to New Zealand and Chile, creating these massive artificial monoculture plantations.
But when you dig up a pine sapling in Europe or North America to ship it overseas, you aren't just taking the tree. You are taking a massive clump of the native soil packed around its roots.
And inside that soil was the symbiotic partner the pine tree absolutely required to survive.
Yeah.
The Fly Agaric, Amanita muscaria.
The timber companies inadvertently shipped the Fly Agaric all over the world.
And because we now know the peppery bolete is an obligate parasite of the Fly Agaric, the chain of events becomes terrifyingly clear. The tree brought the Amanita, and the Amanita brought the Chalciporus.
The parasite followed its host across the oceans, establishing itself deep within these exotic pine plantations in the Southern Hemisphere.
But life finds a way to expand, and the story doesn't end at the edge of the pine plantations. What happened in places like Tasmania, Victoria, in Australia, and across New Zealand is a phenomenal evolutionary leap.
It is. The introduced Fly Agaric didn't stay confined to the rows of planted timber. It executed what ecologists call a host jump.
And a host jump is a significant, often destabilizing ecological event, right?
Very destabilizing. The invasive Amanita muscaria somehow adapted its biochemical signaling to form novel symbiotic relationships with the ancient endemic trees of the region, specifically the native Nothofagus or southern myrtle beech trees.
So it breached the perimeter of the artificial plantation and infiltrated the pristine native ecosystem.
And because the Peppery Bolete is inextricably tethered to the Fly Agaric where the Amanita went, the parasite followed.
The peppery bolete spread from the commercial pine forests straight into the ancient native woodlands, continuing to parasitize the fly agaric in this entirely new habitat.
It's an invisible invasion network.
It's crazy. You have a native southern beech tree feeding an invasive fly agaric, which is simultaneously being bled dry by an invasive peppery bolete. It is a staggering chain of ecological exploitation.
It truly is. However, the hierarchy of parasitism rarely stops there. In biology, an apex predator is usually a temporary concept.
Right.
This brings us to the phenomenon of hyperparasitism. Just as the peppery bolete ruthlessly parasitizes the fly agaric, the bolete itself is hunted by an even more specialized microscopic fungal assassin.
It is the biological equivalent of Russian nesting dolls, each layer consuming the one larger than itself. This microscopic assassin is a highly specialized mold known as Sepedonium chalcipori.
And its naming is no accident.
No, it doesn't attack the beech trees. It doesn't attack the fly agaric. It evolved specifically and exclusively to hunt the peppery bolete.
The pathology of Sepedonium chalcipori is brutal. It is classified as a necrotrophic parasite.
Necrotrophic, so it kills it.
Exactly. Unlike a biotrophic parasite, which tries to keep its host alive as long as possible to siphon a steady drip of nutrients, a necrotrophic parasite's strategy is rapid, overwhelming destruction. When it infects the peppery bolete, it induces widespread necrosis, rapid cellular death.
It essentially melts the flesh of the bolete while it's still standing in the forest. It transforms this firm, peppery mushroom into a gruesome, collapsing mass of golden yellow spores, which mycologists call conidia.
The visual transformation is total. But the truly remarkable aspect of this infection isn't just the destruction of the host. Right. It is the absolute lack of secondary scavengers.
Right, because normally in a forest environment, the moment a fleshy mushroom dies and begins to decompose, it is instantly swarmed. It becomes a chaotic buffet for dozens of competing species of ambient bacteria, environmental molds, and insect larvae.
But observation shows that when Sepedonium infects a bolete, that swarm never arrives. The rotting mushroom is eerily pristine. You almost never see any competing bacteria or rival fungal colonies growing on the carcass.
The Sepedonium mold manages to monopolize the resource entirely. To figure out how it does that, we have to look at the chemistry.
The mold achieves this monopoly through aggressive, highly sophisticated chemical warfare. Once Sepedonium breaches the bolete's tissue, it immediately begins synthesizing and exuding a formidable arsenal of bioactive compounds right into the surrounding flesh.
And the vanguard of this chemical assault relies on a class of incredibly potent antibiotic compounds known as peptibols.
Yes.
Let's unpack the mechanics of peptibols, because this is where the biological engineering gets truly wild. How do these compounds actually function as weapons?
Peptibols are a specific, structurally unique class of oligopeptides, essentially short, specialized chains of amino acids. What makes them so phenomenally lethal to competing microorganisms is their structural composition. They are heavily populated by non-standard amino acids, which most standard cellular machinery just cannot process or defend against.
They're built from a chemical language that the competing bacteria don't even have an alphabet for.
Exactly. And their physical action is devastating. Because of their unique helical structure, peptibols are lipophilic. They love fats.
Okay.
They actively insert themselves into the lipid bilayer, the protective fatty membrane that surrounds and protects the cells of competing bacteria and fungi. Once embedded, these short peptide chains aggregate together and physically form pores.
They literally punch microscopic holes in the armor of their enemies.
Yes.
So a competing environmental bacteria lands on this rotting bolete, hoping for a free meal, and the Sepedonium mold hits it with these peptibols. The peptibols lodge in the bacteria's cell wall, punch a hole right through the membrane, and the bacterial cell loses all its internal pressure, leaks its cellular contents, and dies instantly.
It is a microscopic, highly targeted, localized sterilization. Working in concert with a suite of other toxic secondary metabolites, the peptibols effectively establish an impenetrable, highly toxic exclusion zone around the dying mushroom.
So the Sepedonium mold builds a chemical fortress, allowing it to slowly and exclusively consume the nutrient-dense flesh of the bolete without expending any energy fighting off physical competitors.
It's ruthless, brilliant biological engineering, and this understanding of chemical exclusion zones perfectly sets the stage for the landmark 2021 survey published in the journal Microorganisms.
The paper that takes us deep into the forests of South America.
Yes, this research paper is a vital piece of the puzzle regarding global fungal interactions, and the geographical setting is paramount here. The research team conducted exhaustive field surveys in the Biobío, Dubal, and Araucanía regions of temperate central Chile.
And the landscape here is defined by extreme ecological tension. If you look at aerial mapping of the region, you see a stark, fractured mosaic. On one side, you have massive, rigidly geometric blocks of exotic timber plantations, endless rows of introduced eucalyptus and pine trees planted for the global market.
And pressing right up against the borders of these artificial grids are the fragmented, shrinking remnants of the primeval Nothofagus, the native southern beech forests.
It is an active battleground between the industrialized and the ancient.
The researchers focused their efforts precisely on these transition zones, actively hunting for macroscopic mushrooms showing signs of mold infection, and their initial findings visually confirmed the invasive migration we discussed earlier. They documented a widespread, entrenched invasive network. They found, introduced, non-native boletes thriving in the Chilean soil, including our Peppery Bolete, Chalciporus piperatus, alongside others like Paxillus involutus and Celis luteus.
And just as expected, these invasive mushrooms were heavily infected by globally widespread species of the Sepedonium mold. The researchers identified Sepedonium ampulosporum, Sepedonium chrysospermum, and Sepedonium levigatum.
It was a complete imported ecosystem. The invasive timber brought the invasive mushrooms, which brought the invasive molds.
That finding alone would be a solid ecological confirmation, but as they continued to sample infected mushrooms deeper into the native Nothofagus stands, they made a monumental discovery.
They did. Amidst this sea of globalized invasive fungi, they isolated a morphological anomaly. They discovered a brand new, completely distinct species of mycoparasitic mold that was entirely unknown to science.
And they designated it Sepedonium leorum. And what makes Sepedonium leorum such a massive discovery isn't just that it's new, it's the mold's absolute unyielding host specificity. Remember, the forest is currently flooded with invasive boletes, but Sepedonium leorum completely ignores them.
Completely ignores
It
it.
does not attack the peppery bolete. It does not attack the suillus. It exclusively and aggressively targets only native endemic Chilean boletes.
The precision of its parasitism is astounding. The research team documented F. leorum infecting 4 specific native mushroom species: Boletus loida, Boletus loyo, Boletus putatus, and Gastroboletus valdivianus.
And these 4 mushrooms are endemic to these specific Chilean forests. They exist naturally nowhere else on the planet.
And their dedicated hyperparasite exists nowhere else either. It is a textbook manifestation of a highly specific, co-evolved parasite host guild.
But proving you have discovered a new species in modern biology requires far more than just pointing at a fuzzy yellow patch on a mushroom and saying, I haven't seen this one before. You have to build an ironclad case utilizing multiple scientific disciplines. So how did they prove S. leorum was truly unique?
Well, the methodology starts with rigorous morphological analysis, utilizing both high-powered optical microscopy and scanning electron microscopy to capture the physical structure at the nanometer level.
Okay.
Macroscopically, when S. leorum first colonizes a native bolete, it generates a white tomentose mat. Tomentose simply means it is covered in densely matted, wool-like fungal hairs.
And then it changes color.
Yes. As the infection matures and spore production begins, the entire mass transitions to a striking, vibrant golden yellow.
But the true identity is locked in the microscopic architecture, right? When the researchers look through the electron microscope, what specific shapes tell them they are looking at something new?
They focus heavily on the kinidiophores, the microscopic branching structures that physically bear the reproductive spores. In S. leorum, the architecture of these branches is highly distinct. They form verticils, which are essentially whorled arrangements of branches radiating from a single point, somewhat like the spokes of an umbrella.
Okay.
These verticils bear up to 3 and very rarely 4 short, specialized terminal cells called phylletes. Those are the exact points where the spores are generated.
It's incredible that a mold has such rigid geometric rules for how it builds its microscopic branches.
It really is. The defining characteristic, however, is the morphology of the Illyrioconidia. These are the thick-walled, heavily armored survival spores, designed to withstand harsh environmental conditions until a new host becomes available.
They're pretty big.
Relatively massive on a cellular scale, measuring 10 to 15 micrometers in diameter,
Yeah.
and their surface is entirely covered in densely packed, highly structured warts.
And the paper gets incredibly descriptive about these warts. They don't just call them bumps. Under the scanning electron microscope, these microscopic warts are specifically shaped like tiny barrels, miniature volcanoes, or amphoras.
Amphoras.
Those classic narrow-necked ancient Greek vases, complete with a distinct tiny depression right at the apex. The amount of genetic instruction required to build millions of perfectly shaped microscopic Greek vases on the surface of a single spore is baffling.
It's stunning. But while the morphological evidence, the unique verticils in the amphora-shaped warts is compelling, the gold standard for species delineation today is molecular phylogenetics.
Right.
The physical form gives you a hypothesis.
Yeah.
The DNA gives you the proof. The researchers extracted the genetic material and sequenced 3 distinct, highly conserved DNA markers.
They used the ITS region we mentioned before, but they also added 2 more, RPZ2 and EF1 alpha. Think of these as 3 different, incredibly precise evolutionary clocks ticking away inside the genome.
Precisely. By comparing these 3 specific sequences against the genetic databases of every other known Sepedonium species on Earth, they triangulated its exact position on the evolutionary tree.
And the molecular data was definitive. The genetic alignment demonstrated that Sepedonium loiorum possessed a minimum 3% genetic distance from its closest known relative, which happens to be Sepedonium chalcipori, the very mold that attacks our Peppery Bolete.
3%.
Now, 3% might sound like a rounding error in casual conversation, but in the context of conserved genetic markers like these, what does a 3% divergence actually represent?
In evolutionary genetics, a 3% divergence across these specific baseline markers represents a vast, unbridgeable chasm. It definitively separates them into 2 distinct species.
Wow.
This genetic distance, combined with the extreme host specificity and the unique morphological traits, proves that Sepedonium loiorum is not a recent mutation. It has been evolving in deep, uninterrupted isolation within these South American nothofagus forests for an immense period of evolutionary history.
It has spent millennia perfectly tuning its chemical weapons, its specific peptibles, to overcome the unique biological defenses of those 4 endemic Boletus mushrooms.
Exactly.
Which brings us to the profound and tragic implications of this discovery. We have spent an hour dissecting the hidden lives of microscopic molds and unassuming forest fungi, and it is completely valid to ask, why should you, the listener, care about the fate of a golden yellow mold in a remote Chilean forest? What does it actually matter?
It matters intensely because Sepedonium loiorum is a masterclass in the fragility of interconnected biological webs. It perfectly illustrates how an ecosystem functions like a Jenga tower.
That's a great way to put it.
And right now, the foundational blocks of that tower are being rapidly removed. The native endemic boletes that S. loiorum absolutely requires for its survival are facing an existential crisis.
The conservation data is bleak. According to the IUCN Red List of Threatened Species, the Global Authority on Extinction Risks, Boletus loio and Gastroboletus valdivianus are officially classified as endangered. Boletus loida carries a vulnerable status, and Boletus putidus is near threatened.
The only organisms on Earth that this newly discovered mold can eat are actively disappearing.
This scenario introduces one of the most alarming and often overlooked concepts in conservation biology: coextinction.
Right.
When we think about extinction, we usually picture a single species being hunted or losing its habitat, but highly specialized parasites face a double cascading threat. They are directly vulnerable to ambient environmental changes, yes, but they are inextricably, fatally tethered to the population dynamics of their hosts.
It is a terrifying domino effect driven by human industry. Let's trace the collapse. Anthropogenic land use changes, specifically the aggressive clearing of ancient land to plant more rows of exotic, fast-growing timber for the global market, destroy the native nothofagus beech trees.
And without the root systems of those specific beech trees, the native Boletus mushrooms lose their symbiotic partners and die out.
And if the native Boletus goes extinct, the highly specialized Sepedonium loiorum mold, completely incapable of digesting the invasive mushrooms that replace them, goes instantly extinct right alongside them.
A single logging operation doesn't just cut down a tree. It can simultaneously wipe out 3 distinct, interconnected trophic levels of endemic species in one fell swoop.
Habitat loss through logging is the blunt force trauma to the ecosystem. But the research points to a secondary, much more insidious threat operating below the surface: the invasive wave.
Right. We established how the invasive fly agaric, Amanita muscaria, has breached the boundaries of the pine plantations and is actively making host jumps deep into the pure native Nothocus forests.
And we know exactly who is riding the Fly Agaric's coattails into that native territory. Our heavy metal-absorbing, peppery-tasting friend, Chalciporus piperatus.
And right behind the Peppery Bolete, utilizing it as a biological Trojan horse, are the invasive Sepedonium molds like S. amplosporum and S. chrysospermum.
This invasive front is carrying an exotic, highly evolved arsenal of chemical warfare directly into the heart of an ancient, isolated ecosystem. We have absolutely no data on how the native fungi will react to these foreign biological weapons.
Will the aggressive, globally adapted invasive moles outcompete the highly specialized endemic S. loyorum for spatial territory?
More concerningly, could the specific exotic peptiboles deployed by these invasive molds prove lethal to the already endangered native Bolete species?
The potential for total ecological replacement is massive and mostly invisible to us.
Yeah.
We could lose an entire ancient fungal ecosystem, millions of years of isolated coevolution, to a silent microscopic chemical war happening right beneath the leaf litter.
We risk replacing a unique endemic tapestry of biodiversity with a homogenized, globally invasive suite of fast-spreading fungi.
It serves as a profound, urgent reminder that comprehensive conservation cannot solely focus on charismatic megafauna, the pandas, the whales, the tigers. The foundational health of our planet is dictated by these microscopic, highly specialized ecosystems.
The loss of a single, localized hyperparasite like S. loyorum represents the irreversible erasure of unique chemical and evolutionary history.
Let's pull all these threads together because the journey we've taken based on these sources is staggering. We started with the Peppery Bolete, Chalciporus piperatus, a mushroom that frustrates chefs by tasting like intense black pepper raw, only to completely lose its heat the second you cook it to neutralize its toxins.
We discovered its inexplicable ability to act as a biological sponge, thriving in devastated, heavy metal polluted industrial wastelands by somehow safely absorbing thousands of milligrams of lethal antimony.
We then dismantled decades of mycological assumptions, utilizing isotopic forensic tracing to reveal that it was never a helpful symbiotic partner to the forest canopy, but rather a ruthless mycoparasite silently siphoning nutrients from the famous, globally recognized Fly Agaric mushroom.
We tracked its incredible oceanic migration via the commercial timber industry, watching it adapt and jump from introduced pine trees deep into the ancient native forests of the Southern Hemisphere.
And ultimately, we zoomed into the microscopic level of hyperparasitism. We examined the brutal Sepedonium molds that melt these boletes into yellow goo by deploying pore-forming antibiotic peptibles to slaughter any competing bacteria.
We traveled to the fractured, endangered Nothofagus forests of central Chile to witness the discovery of Sepedonium loyorum,
A newly documented hyperspecific mold that creates microscopic amphora-shaped spores standing on the absolute brink of coextinction alongside its endangered native hosts, threatened simultaneously by human logging and the creeping silent invasion of the very fungi we started this journey with.
The overarching narrative perfectly illustrates that an entire world of unimaginable complexity, relentless chemical warfare, and profound ecological fragility exists continuously just out of our normal field of vision.
Which leaves us with one final, incredibly provocative thought for you to mull over. We know, based on the research, that these Sepedonium molds have evolved the ability to synthesize these incredibly powerful, complex antibiotic peptibole compounds capable of physically punching holes in the cellular armor of bacteria to ward off all pathogens.
And we know for a fact that the Peppery Bolete can somehow chemically isolate and absorb thousands of milligrams of toxic antimony without suffering cellular death.
The biochemistry actively occurring in the dirt beneath our feet is vastly more intricate, efficient, and advanced than anything we can currently synthesize from scratch in a pristine laboratory.
So consider this. What if the key to our next great medical breakthrough? What if the next iteration of penicillin, the exact molecular structure we desperately need to defeat the rising tide of antibiotic-resistant superbugs? Or the precise biological chelating agent required to safely and cheaply clean up our most toxic, heavy metal polluted Superfund sites, isn't waiting to be discovered in a multi-billion-dollar pharmaceutical lab?
What if the answer is currently hidden inside the microscopic chemical warfare of a hyperparasitic mold actively feeding on a spicy, toxic mushroom deep within an endangered, rapidly shrinking South American forest? And if human industry causes that forest to disappear before we can fully decode its biology, what miraculous, world-changing cures simply vanish with it?
What other invisible, high-stakes chemical wars are happening right under your boots right now, holding secrets we haven't even begun to imagine?
It is a profoundly humbling reality, and perhaps the most powerful argument for aggressively protecting even the most unseen, seemingly insignificant corners of our natural world. We hope this deep dive has completely rewired how you view the forest floor. The next time you walk past a simple brown mushroom, remember the incredible, violent, and complex machinery operating silently just below the surface. Stay endlessly curious, keep asking the hard questions, and we will see you on the next deep dive.